class, and welcome to Chapter 1 of Biological Psychology. So, this lecture is on Chapter 1, which covers nerve cells and nerve impulses. I know it's a little dark in here. I will switch the lights on when I switch over to my whiteboard to draw my demonstrations for neuronal firing, but I did this way so you won't have a glare from the screen from the lighting. So, we will begin Chapter 1 with the mental experience. So basically, for this entire semester, we're going to be covering how the neural system, so basically the central nervous system, which is made up of the brain and the spinal cord, how they work together to create the mental experience. What is the mental experience? Basically is your everyday life. It's the recording of your memory, your how-to, your feelings, emotions. How is it that you're able to listen to me right now and your body is able to calculate the temperature at the same time? It's understanding whether you're hungry or not. It is proliferating cells at the same time. You are able to understand me as well as hear me and also take notes. The mental experience is basically your whole waking life, the life that you consider to be conscious. How does the central nervous system create the mental experience? It boils down to these two cells, the neurons and the glia cells. You have over 100 billion neurons, and you have even more glia cells. So glia cells are basically like the helper cells to the neurons. They kind of go between to orchestrate various events, which we're going to talk about because there are various different types of glia cells that helps the neurons. There are several types of neurons. The ones that we're going to be talking about in this class throughout the entire semester are the motor neurons, the sensory neurons, and the interneurons. Don't worry, at this point, we'll get to that slide when we talk about the neurons more specifically. But as for right now, we are going to discuss the neurons, how the glia cells help the neurons, and how the neurons how they work together to create the mental experience. Your central nervous system, which is made up of the brain and the spinal cord, are made up of billions of neurons that work together to create your mental experience. Okay, so with that being said, we'll move to the next slide. Okay, I have this clip just in case you were not aware of how the neuron actually goes about firing. So it's kind of like an action clip. Let me go back. Let's click on here. I can't, there it is. So this is the brain of the central nervous system, and it just shows how it's broken down into these neurons. As I said before, we have over 100 billion neurons, and they fire. So the firing are basically electrochemical nerve impulses that they send from one neuron to the next. So basically what you're seeing here is a demonstration of how the neurons actually send electrochemical nerve impulses to each other. So this is how they communicate. Okay. Let me turn this off before it goes to the next one. Okay. It won't let me. There we go. Okay, so some neuroanatomy. So the neuron is this structure here. Okay, I hope you guys can see that. I'm holding my computer, obviously, trying to keep it steady. Okay, so the neuron is made up of several different components. It starts off with the branching tree-like structures, which are called the dendrites here. In the middle of the neuron, you have this circle here, which is the soma. The soma houses the nucleus. The nucleus, if you guys remember from biology, it houses all of the organelles, the essential organelles, so the ribosomes, the mitochondria, endoplasmic reticular, all those organelles are housed within the nucleus. So the nucleus, the little yellow dot, houses, the DNA is also housed within the nucleus. So the nucleus resides within the soma. So you have the dendrites, which is the branching tree-like structures, and then you have the wider area, redder area, which is the soma. And within the soma, you have the nucleus. Then it branches off into this long nerve fiber, which is called the axon. Okay, the axon is then wrapped with myelin sheaths, which is known as myelination. And then you have at the end of the axon, you have the presynaptic terminals, which are also known as butons, B-U-T-O-N-S. Okay, so again, we have the dendrites, the cell body, the axon, the myelin sheaths, and the presynaptic terminals. This is what the neuron looks like under the microscope. Okay, one fascinating detail, information to know about the neurons is that they communicate, but they do not touch. Okay, so they literally transfer electrochemical information from one neighboring neuron to the next without touching each other. Okay. So now we're going to go over each of these structures separately to talk about their functionality. Okay, so let me back this up a little bit. The dendrites. So the dendrites, think of them as branches from a tree. The job or the function of the dendrites is to bring in electrochemical information from the neighboring neuron. So these are the dendrites. This is what a real one looks like under a microscope. They branch out. They are covered with synapses or synaptic receptors. Okay, and if you think of, you can think of the synaptic receptors as satellite dishes. So if you think about your cable company, in order for you to receive the information, they put a satellite on the side of your house. So those satellites bring in, or they receive information from the mother satellite in outer space. Okay, so the synapses essentially acts as the same way. They act in the same way. They function the same way. They're covered. You see these little bitty dots, little bitty red dots. Those are synapses or synaptic receptors. They cover all of the dendrites, and they act as satellite dishes in order to bring in information from the neighboring neuron. So the job or the functionality, the basic function of the dendrites is to receive electrochemical information. Okay, this is what it kind of looks like up close. So basically what you see are the gray branching structure are the dendrites, and these are the ends of a neighboring axon, which are the presynaptic terminals. So if the presynaptic terminals are facing the dendrites, it means that it is sending information into the neighboring neuron. So the presynaptic terminal are the ends of the neuron, and the dendrites are the beginning of a new neuron. So the presynaptic terminals are sending electrochemical information into the dendrites. So the dendrite's job is to receive. It receives this information from the neighboring neuron. Again, this information is known as electrochemical transmission. Okay, so again, the job of the dendrites is to receive electrochemical information. The soma. So here are the dendrites again. And then you have that little round part in the middle that I was showing you here. The round red circular area is the soma, which means cell body. Okay, and it houses the organelles. I have this slide up here just to show you how tiny the soma is. So if you see here, the soma ranges between 0.005 millimeters to 0.01 millimeters in diameter. A sharpened pencil is 1 millimeter in diameter. So this is way smaller than the tip of a sharpened pencil. I just wanted to put this up here so you'll get an idea of just how small these neurons are. So they're really, really tiny, or super tiny. And they're all compacted within your brain in order for it to operate, to create a mental experience. So the soma, again, the soma houses the organelles of the cell, okay? Mitochondria, the ribosomes, the endoplasmic reticulum. And it also houses the DNA, okay? So the soma, which is the cell body, houses the nucleus. The axon is a long branching structure with different lengths. So not all axons have the same length. Some are longer than others. Some are very short. Some are very, very long. It just depends on the job or the function of that particular neuron. Okay. So if you notice, we have the long branching structure, and then we have this green looking structure that wraps around the entire axon. This is myelination. It's called the myelinated sheaths. Okay. The myelinated sheaths helps to speed up the electrochemical transmission. Okay. So once the dendrites receive the information, its job is now to transmit. The other parts of this neuron is now to transmit, works together to transmit this electrochemical information from one end of the dendrites to the next, to then send that information to the next neuron. So the job of the myelin sheath that wraps around this axon here, which is a nerve fiber, is to speed up the neurochemical information from one end of the axon to the next. So the myelin speeds it up. It's made up of fatty tissue, fatty cells, and it wraps around the axon. And its job is to speed up the chemical information from one end of the axon to the next. Okay, so once the axon fires, it goes pow, shoo. And what makes it go fast, again, is the myelin sheaths. If you notice, there are gaps in between the myelin sheaths. So there's a gap here, and there's a gap there. The myelin sheath is not an entire whole structure. There are gaps in between. The gaps in between the myelin are known as the nodes of Ranvier. Okay, well, I spelled it here. The node of Ranvier. Some people pronounce it Ranvier or Ranvier if you want to be fancy. But the job of these spaces in between we'll talk about later. Okay, because a lot happens from node to node in order for the electrical chemical information to get from one end of the axon to the next. So these nodes of Ranvier, the spaces in between, are very important. Okay, but I'll stop right there when it comes to the node of Ranvier, and we'll visit that later. Then we have efferent and afferent. So some axons are afferent, and some axons are efferent or efferent. Okay, so as I said before, we're going to talk about 3 different types of neurons. Okay, I hope you're looking at your slides as you're looking at this and following along. So we have the sensory neurons, we have the motor neurons, and then we have one in the middle that are known as the interneurons, or the intrinsic, also known as the intrinsic neurons. Okay, so the sensory neurons are known as the afferent neurons. They bring in information into the central nervous system. So what do I mean by that? Think of the 5 senses, the 5 physical, so I'm not speaking of the 6th sense. I'm talking about the 5 physical senses. So hearing, tasting, smelling, touching. I think I've said all right. Hear, taste, smell, touch, seeing. Okay, sight. Those neurons are bringing in information into your brain. Okay, all of your 5 senses are separate, or they bring in information separately and specifically for each sense, and it brings that information into your central nervous system. Again, your central nervous system is made up of your brain and your spinal cord. Okay, so all of your senses bring in information into the central nervous system. Okay, there are senses. So your 5 senses, the neurons that governs your 5 senses, all of them are located in the peripheral nervous system. So far I've just mentioned the central nervous system, which is the brain and the spinal cord, but I have not mentioned everything else. So everything else outside of the central nervous system is known as the peripheral nervous system. Okay, So the job of your sensory neurons from all of your 5 senses is to bring in sight, bring in taste, bring in smell, bring in auditory information into the central nervous system. So your senses tell the brain what's going on in the outside world. Okay? So they are known as afferent. Afferent. They bring in, they admit. Another way of thinking of this as A is for admit and E is for exit. We haven't talked about efferent yet. Afferent is admit. Your senses bring in information into your central nervous system. Okay? That's how your brain knows what's going on in the outside world. Okay? Efferent is when your brain has integrated the information that it has received from the afferent neurons, and it has now taken that information, consolidated it, gotten instructions from your motor system, and is now sending information, commands to your muscles and the rest of your peripheral nervous system in order to carry out those commands. Okay, so those are afferent. Your motor neurons are the efferent or efferent information system. Your efferent or efferent information is that, which is the information that has been integrated in the brain and now sent through the motor output area to the muscles in the peripheral nervous system. So again, afferent sensory neurons bring in specialized information into the brain to let the brain know what's going on the outside world. The brain integrates it. How does it integrate it? Through the intrinsic neuron. The intrinsic neurons are found mostly in the spinal cord. Okay, so the intrinsic neuron help to connect the sensory neuron to the motor neuron. So sensory bring in from the 5 senses. They're coming in, information coming in from the 5 senses, and then it integrates via the intrinsic or the interneuron. This little neuron here is either known as the intrinsic or the interneuron. Okay, it has a very short axon. So its dendrites and cell bodies, as well as its presynaptic terminal, are within the same structure, which differs for the sensory neuron, which can have its dendrites in the eyes and its ending presynaptic terminals in the occipital lobe. Same for motor. So the intrinsic or the interneurons are very short neurons that connect the afferent sensory neurons to the efferent or efferent motor neurons. So again, sensory information goes in, which are afferent, carries information into the central nervous system, integrates this information, and then the brain will send motor commands to the muscles. So it exits that motor commands exits the central nervous system and is sent out to the peripheral nervous system, which is where you will find your muscles. Okay, so just from the diagram, this person is looking at the glass of water. So he's using sight information. Not only is he using sight information, this sight information is very important because it's calculating how much water is in the glass. Also, how much force, the brain is also calculating how much force it needs to, force information it needs to give your muscles in order to pick this glass up without just dunking it in your face, as babies do when they pick up their first glass of water to try to drink it. So sight information is this information, integrating it into your brain and your spinal cord is very important because it's calculating how heavy this glass is, how much force you have to use to pick up this glass of water, okay, without just picking it up and dunking it all in your face, okay? So afferent is bringing in information into your central nervous system. Your central nervous system says, okay, thank you, we're going to calculate this. This is what you have to do in order to drink this water. So it sends out that motor command to your muscles, efferent. Input information in, afferent. Exit motor information, efferent. Okay. So the node of Ranvier, I got ahead of myself. Okay, so the gaps in between the myelin, as I said before, are known as the node, you can see it better here, are known as the node of Ranvier or Ranvier. Okay, And in between, which they are made up by the gaps in between the myelin sheaths along the axon. Okay, so I have up here, it's responsible for saltatory conduction. Saltatory means to jump. So when you looked at the video, it looked like the electromagnetic waves were just electromagnetic waves. The electrochemical nerve impulses were transmitting smoothly from one end of the axon to the next. But actually, when you look at it under a microscope, it looks like it's jumping from node to node. So when it fires, it is a electrical combustion that starts here at the axon hillock, which is the axon hillock. This is not in your book, but the axon hillock is where your soma branches off into the axon. So this is this wide area here where it starts to branch off into the long nerve fiber, which is called the axon. It's called the axon hillock. So the firing starts at the axon hillock, and it sends a nerve impulse from node to node. So at every node, there's a new firing of the neuron in order for the electrochemical information to get from one axon, end of the axon, to the next without fizzling out or losing its power. Okay. This is what the axons look like under a microscope here. And you'll see like the little nodes of Ranvier. This is the breaking of the myelin sheaths. So the darker parts, this is one long axon. And it's dark because it's wrapped with the myelin sheath. And where the myelin sheaths end and where it begins is the little gaps called the nodes of Ranvier. So the nodes of Ranvier literally are the gaps in between the myelin. And there's a whole nother part to this lecture today that we're going to cover that shows you what actually happens step by step at the nodes of Ranvier. The presynaptic terminal, okay? So as I was saying before, the neurons communicate, but they don't touch. Okay, so if you see here, this is the one end of the axon, which is where the presynaptic terminal branches out into like a button. Think of it as the elephant's foot. Okay, it kind of balloons out. So this is the axon. So if we started at the dendrite, so say for instance, we can see the other part of this axon, which would be the dendrite. So we have this long branching axon. We've gotten to the end of this axon, which is the presynaptic terminal. Okay, it faces or it synapses onto the dendrite of a neighboring neuron. So every presynaptic terminal is facing the dendrite of a neighboring neuron. This is how they send information from one neuron to the next. Okay, there's a whole 8-step process that we're going to go over later in this semester for you to understand how this happens, this process of them releasing the neurotransmitters into the synapse. Okay, but for right now, this is just anatomy. We're just going over the functional parts of the neuron. So the presynaptic terminal buttons or boutons. I spelled the boutons version earlier, or you can just call them the buttons. Okay, and they just call it that because they kind of balloon out like buttons at the ends. Okay, so the presynaptic terminals are where the neurotransmitters are housed. If you see these little spherical sacs inside of the axon, the end of the axon, which is the presynaptic terminal. Okay, and then they are released into the synapse and they lock on to the receptors of the neighboring dendrite in order to activate this dendrite to tell it to activate and further send this information down its axon to the neighboring neuron. Okay, so there's a whole lock and key method we're going to discuss later in this semester. Okay, this is another diagram. I use a lot of diagrams just because I have different types of learners in the class. But again, this is the axon, okay? And then at the end of the axons, you have the red presynaptic terminals or the buttons. And they're facing the dendrites of a new neuron because the job of the dendrites is to receive electrochemical information. The job of the presynaptic terminal is to release or send the electrochemical information through the neurotransmitters that it releases. Okay, so the neurotransmitters we haven't talked about yet, but they lock on to the dendrites in order to activate this neuron. So the job of the dendrites, again, is to receive this information. And the job of the presynaptic terminal is to release, to send this information to the dendrites. Okay, so types of neurons, we kind of discussed this already. I got ahead of myself again. But the types of neurons are the motor neurons, which I was saying earlier that they were afferent. Okay, the motor neurons are located within the spinal cord. Okay, motor neurons are located within the spinal cord, and there are other structures that connect onto this, which sends out axons to the other peripheral areas, such as the muscles, and innervate the skin and other organs within your body. Okay, so everything outside of the brain and the spinal cord is the peripheral nervous system. Okay, this here, brain and the spinal cord, is the central nervous system. Okay, think of it as a computer. So your CPU, central processing unit, would be likened to the brain, your brain and the spinal cord. Okay, and the keys on the keyboard are the peripheral. So if you input information, you input a certain combination, which would be a code, the brain would then integrate this information. Okay, so these keys would be likened to the sensory neurons inputting this information into the central processing unit, which in relation to us, it would be the sensory input. We talked about this, how this is afferent. It admits this information into the brain and the spinal cord, which is the central nervous system. Okay, and then once the computer integrates this information, then it sends out commands. It carries out those commands, and it sends it through the motor axons to the muscles in order for you to respond to whatever task that you're trying to complete. Okay, so if you type in your email, or if you type in Gmail, you press enter, it's going to give you what? The Gmail website. Okay, your body functions the same way. Okay, just like here in this diagram. So let me show you, see if I can focus it better. That should be better. You guys can see that you have the finger is too close to the flame. Okay, heat, which is the sense of touch, is picked up by the sensory, the touch receptors embedded within the skin. It brings in this information to the central nervous system, which is the brain and the spinal cord. Okay, it integrates this. This is this little white structure here, if you guys can see that. That's the intrinsic, or which is also known as the interneuron. It connects the sensory information to the motor. Okay, so this sensory information of heat is transferred to this interneuron or intrinsic neuron, where it integrates. Okay, again, this is in the spinal cord. So the central nervous system is made up of the brain and the spinal cord. It sends this information once the information is being processed, it sends the command information, the motor command information, to the peripheral, which is located specifically the muscles that are in your arm, in order for this person to move his hand away from the flame so they won't become burned. So that would be an example of efferent. So afferent is admitting sensory information from this heat to the intrinsic neuron in the spinal cord. Spinal cord integrates this information, says, oh my gosh, we're about to burn, it's too hot. Let's move our hand away. Sends that motor command to your muscles and the peripheral nervous system for you to move the hand away from the flame. Okay, so there's always an ongoing input-output informational processing that's going on between your sensory and your motor by way of the intrinsic and your interneurons. Glia cells. We have five glia cells that we're going to discuss in this class. One of them being the astrocytes, the other the microglia. The other one is the oligodendrocyte, and its cousin is the Schwann cells, and then we have the radial cells. So let's do the oligodendrocytes and the Schwann cells first, because they are the easiest. Okay, oligodendrocytes. oligodendrocytes and the Schwann cells perform the same function. They both create the myelin that you see or that you saw wrapped around the axons. Now, I should say that all axons are not myelinated. There are some axons that have no myelination. Also, if you have some type of disorder such as, what is the name of that? I almost called it. What is the name of that disorder? It'll come to me. It'll come to me. But if you have some type of disorder that attacks the myelin, what is the name of that disorder? It skips me at the moment. But there is a disorder whereby the myelin, the cells, the bacteria, antigens, pathogens will actually attack, or they'll eat the myelin wrapped around the presynaptic terminals. That will stop down the neurochemical transmission, which basically leads to slower cognition. So you think slower, you move slower. It almost came to mind. It'll come to me later. But anyway, so the astrocytes, their job is to wrap around the presynaptic terminals, and they do a multitude of tasks. One being the breakdown and the reuptake of the neurotransmitters, which we'll talk about later. But once the neurotransmitters are released into the synapse, which again we have not talked about yet. So basically, the neurotransmitters have been released from the presynaptic terminal. Multiple sclerosis, that's it. I was trying to think of that disorder, multiple sclerosis. So in individuals who have or been diagnosed with multiple sclerosis, it attacks the myelin sheaths that are wrapped around the axons, which causes slower cognition, movements, speech, what have you. Okay. Most people who have been diagnosed with multiple sclerosis don't really know how their body is going to be attacked from day to day. So it's kind of random, but for the most part, it eats away at the myelin that's wrapped around the nerve fibers, which are the axons. So, as I was saying, the neurotransmitters that have been released from the presynaptic terminals, once they have been released, they have to clear out the passageway. So that gap in between the presynaptic neuron and the postsynaptic neuron, which is the neighboring neuron. So the job of the astrocytes is to break down those neurotransmitters and help it to go back up into the presynaptic terminal that has just released it. I'm just mentioning this right now. We're going to go more into depth later on in the semester for you to understand how this happens more specifically. It also, so the astrocytes also govern the synchronicity of the firing of the neurons. So it acts as a stop and go traffic signal. So one set of neurons are firing, which are basically sending electrical chemical information from one neuron to the next. Another set have stopped firing. So it's like you fire, okay, now this set fire. Okay, now this set stop. Okay, now this set fire. It synchronizes the firing of the neurons. So they won't all fire at once. If all your neurons were to fire at once, you can actually have a seizure. If you look this up, you can go to Google. You'll see that one cause of epileptic seizures is or a lack of astrocytes in the brain. Okay, so one job is for the astrocytes is to synchronize the firing of the neuron. Another job of the astrocytes is to remove waste material. So neurotransmitters that have been broken down or have undergone reuptake process can leave debris within the synapses between the neurons, and that can happen. So the astrocytes actually engulf the waste, engulf or digest this material in order to clean the leftover debris from the breakdown out of the synapses. Then you have the dilation of the blood vessels or the blood capillaries. So when the brain is processing, so say for instance, I have someone to conduct mental arithmetic in front of class. Okay, so in order for that person to achieve this function, the frontal lobe has to integrate with the parietal lobe and the occipital lobe. So what you'll see, if I had a fMRI, which is functional magnetic resonance imaging machine, it will show blood flowing, more blood increasing in the areas of the frontal lobe, the parietal lobe, and the occipital area, in order for you to solve whatever equation that I gave you mentally. Okay, so in order for the blood to get to these areas of the brain, you have the astrocytes, which dilates those blood capillaries. It'll dilate these blood capillaries for the blood to rush to the necessary areas, brain areas, in order for you to carry out various functions. Okay. So the oligodendrocytes and the Schwann cells do the same job. They create the myelin that wraps around the axons. The only difference is the oligodendrocytes are located within the central nervous system. So they myelinate the axons within the central nervous system, and the Schwann cells myelinate the axons within the peripheral nervous system. Okay. We have the microglia cells. The microglia cells acts as the immune system of the central nervous system. So they further remove waste and help to kill off antigens that have entered the central nervous system, which is the brain and the spinal cord. Then you have the radial cells. The radial cells, we'll talk about this when we get to the brain development chapter. So basically, they help to guide newly proliferated neurons to its destination. So say, for instance, you have newly proliferated neurons and they need to know where to go in the brain. They can follow the radial cells as a guide to the frontal lobe, the occipital lobe, the parietal lobe, anywhere, temporal areas. And once it gets to its destination, once the radial cells have finished guiding these neurons to its final destinations within the brain, then they themselves can differentiate into an oligodendrocyte or an astrocyte, or whatever the brain actually needs. Okay, as far as neurons and glia cells go. Okay. I have these stop and check PowerPoints for you. I do this in class when I'm on campus. But basically, this is just to help you to jog your own memory, see if you have retained what we've gone over so far. So you can pause this video right now and answer these questions on your own. If you cannot answer these questions, you can always go back and revisit this information in the lecture, or you can revisit it in your text. Okay. The videos do not replace reading the text, so you do have to read the chapters. The blood-brain barrier. So within your brain, you have blood capillaries all over the actual brain. And it has to have an extra barrier of protection against entering antigens, pathogens, bacteria, viruses, for example. So your blood capillaries, which are housed within your brain area, they have a special insulation called endothelial cells. So this is a blood capillary here, okay? Your blood capillaries are permeable. They're permeable to different things that the brain needs, like oxygen, carbon dioxide, water, other fat-soluble molecules, nutrients, for example, can pass through various channels to get into the brain in order to nourish your brain. But some things cannot pass through. Well, it's hard for some things to pass through, such as viruses, antigens, pathogens, bacteria. So what keeps them out, or what helps to keep them out, are what are called endothelial cells. So basically, I don't know if you guys can see this. You probably can see this better on your end on your computer, but they are individual cells that are housed tightly. They're compacted very tightly together in order to keep out invaders from entering the central nervous system. Okay. But these you have various other channels. They could be active transport, passive transports. whereby other molecules, nutrients, water, oxygen, carbon dioxide stuff can flow through these to get past these endothelial cells. Okay, active transport. Sorry, someone has a loud motor outside. Active transport uses ATP, which is adenosine triphosphate, in order to actively, which basically is energy, in order to actively transport certain molecules within the blood-brain barrier. So you have active transport, which actually uses energy, which is ATP, adenosine triphosphate, or you have passive ones, which doesn't use energy. It's just kind of open, but only certain molecules that have affinity for this transport can go through it in order to nourish your brain. So the blood-brain barrier basically are the endothelial cells that wrap around the insides of the blood capillaries in order to keep out antigens, pathogens, bacteria. Okay. So how do the neurons fire, which is the second part of this lecture. I'm going to pause right here and turn the light on, and we will reconvene in a second.